We're going to begin another episode of BioTherapy Live. We are live now, whether or not you're watching us in a recorded session. This is a special meeting. They're all special meetings, aren't they? This special meeting is really a continuation of our travels around the globe, looking at biotherapy from practices and research going on all over. We are delighted to have as our guest today Dr. Daniele Dallavecchia, who will be speaking in just a few minutes. But first, Albert is going to give us our classic introduction. Go ahead, Albert. Today's guest speaker is Daniele Dallavecchia, who holds a PhD in biomedicine with a specialization in microbiology from the Faculty of Medicine of the University of Porto, as well as a master's degree in biological sciences from the Federal University of the state of Rio de Janeiro, and a bachelor's degree in nursing from the Alfredo Pinto School of the Federal University of the state of Rio de Janeiro. She is currently a visiting researcher and collaborator at the Laboratory of Dipteran Studies, where she works on research on natural antimicrobials, namely on the action of biochemicals derived from the excretions and secretions of Diptera, or flies, and their effects on bacteria and fungi. Her language skills include Portuguese, Spanish, and English, and probably much more than that too, because her studies have taken her to programs in Brazil, Portugal, United States of America, Denmark, and Switzerland. Dr. Dallavecchia's published papers include original research into the production, rearing, and application of medicinal maggots and how their secretions kill bacteria, fungi, and other microorganisms. Her talk today will address one of her more recent published studies, the biological behavior of the blowfly Chrysomya praetoria after refrigeration, along with the implication for distribution logistics of medicinal maggots. We're also joined by her colleagues, but in the interest of time, I will let her introduce them herself. So with that, I turn the podium over to our esteemed guest, Dr. Dallavecchia.
Thank you so much, Dr. Sherman, the invitations to this webinar. It's a great honor to be here today. I know Dr. Sherman for a long time, but I finally having the opportunity to speak. And I would like to take the opportunity to present the other authors Dr. Alexandre Silva, Alexandre, and Doutora Valéria Aguiar. Dr. Renato cannot come today. And here by my side is my husband, Bernardino, and he will help us with the translations if necessary. Thank you, Dr. Sherman.
Hello everyone who is watching this presentation. Welcome, I'm Dr. Daniele Dallavecchia, and today I will talk about a work that is an important step for the storage and transport of the larvae used in maggot debridement therapy to the site of application. This work studied the biological behavior of Chrysomia putoria after refrigeration, logistics for use in biotherapy. The experimental part of this study was realized at the Diptera Studies Laboratory of the Department of Microbiology and Parasitology of the Biomedical Institute of the Federal University of the State of Rio de Janeiro, Unirio. It was initially founded by the National Council for Scientific and Technological Development, CNPq, and then by the Coordination for the Improvement of Higher Education Personnel, CAPES. Was also part of this partnership the Faculty of Medicine of the University of Porto, where I finished my PhD. This article was published in 2023 in the Journal of the Brazilian Academy of Sciences. Before addressing the main subject of this presentation, I would like to introduce the other collaborators who have contributed to this work. I won't introduce myself as Dr. Sherman has already done so. Renato Silva Filho holds a PhD in sanitary surveillance from the Oswaldo Cruz Foundation, a master's degree in sciences, microbiology from the Federal University of Rio de Janeiro, and a degree in medicine from Unirio. He is an associate professor of microbiology at Unirio and conducts research in microbiology, public health, and entomology. Alexandre Silva holds a PhD in statistics from the Federal University of Rio de Janeiro, a master's degree in statistics from the University of São Paulo, and a bachelor's degree in statistics from the São Paulo State University. He is an associate professor at the Department of Quantitative Methods at Unirio, where he conducts research in applied statistics and educational statistics. Valeria Aguiar has a PhD, master's, and bachelor's degrees in parasitology from the Federal Rural University of Rio de Janeiro. She is a full professor of parasitology and a member of the graduate program in biological sciences, Neotropical Biodiversity at Unirio. Her research focuses on parasitology with an emphasis on entomology. In our study, aim to assess the biological development parameters of Chrysomia putoria fly species. We examined various parameters, including larval, pupal, and total viability, body mass weight of larvae, and the development of insects after storing them at low temperatures for different periods, 12, 24, 48, and 72 hours. Additionally, we analyzed the sex ratio of this species of insects. Refrigeration and larval therapy. Why study this step of the process? Larval therapy is a simple process, but its logistics are not simplified. The study of refrigeration in living organisms, including larvae, is important to understand how different species adapt to low temperatures. This may have implications for larval conservation, as well as understanding what the extreme conditions are for the limit of viability. This is essential to ensure the quality of biomaterials used in larval therapy. Therefore, storage logistics seeks to assure a cold chain with knowledge of the maximum storage time and supportable temperature for biomaterials, which is crucial to assure their viability during distribution and transport for use in biotherapy. And now, I would like to remind you of some benefits of larval debridement therapy, also popularly known as maggot therapy or biotherapy. But first, I would also like to talk about the problem of chronic wounds. Chronic wounds arise predominantly due to an underlying pathophysiological problem, and in these wounds, the normal healing process is interrupted, typically in a prolonged state of inflammation that acts as a microbial substrate environment, leading to the accumulation of cellular debris on the wound surface and causing damage to surrounding tissue, generating infection and necrosis. For many people, maggot therapy is the only hope for these chronic wounds. This therapy consists of applying previously disinfected larvae to the wound bed. The larvae immediately begin their meticulous surgical process with three main phases: debridement, disinfection, and healing. Debridement is the removal of cellular debris and necrotic tissue from the wound. This removal of necrotic tissue eliminates many bacteria that cause biofilm or infection. The larvae release into the wound bed excretions and secretions, which contain a potent mixture of proteolytic enzymes rich in antimicrobial substances. Larvae excretions also release ammonia, causing a change in pH from acidic to alkaline, which is beneficial as most bacteria cannot tolerate this high pH. This is the beginning of the disinfection process. The maggot crawl about the wound facilitates the penetration of these secretions into the necrotic tissue, causing it to break down and liquefy into a nutrient-rich fluid that the larvae ingest. There is a reduction in the microbial load and consequently a reduction in odor. This is a fundamental step to allow the healing process to begin. In this small scheme, we were able to observe that biological debridement carried out by maggot also acts physically, through the hooks that tear the tissue and through the crawling of the maggot that drags cellular and chemical debris, through larval excretions and secretions. Finally, a complete debridement. And now let's take a closer look at what happens inside the wound when the maggot are working at the microscopic level. In the inflammation phase, inflammatory cells are recruited using chemical mediators released into the bloodstream. In this phase, neutrophils and monocytes are recruited from the circulation. As debridement increases and gives space for new tissue to develop, the wound becomes less pro-inflammatory and more anti-inflammatory, which differentiates pro-inflammatory monocytes into anti-inflammatory macrophages. There is an increased expression of vascular endothelial growth factor, VEGF, responsible for the initiation of angiogenesis. In the fibroblast proliferation phase, there is an increase in growth factors, as well as keratinocytes in the basal layer of the epidermis, which will contribute to the growth of epithelial tissue, and there is also an increase in granulation tissue. At this stage, the newly formed blood vessels are deeper and transport oxygen and nutrients to aid tissue growth. In the wound remodeling phase, an increase in fibroblast migration is observed, and the extracellular matrix is able to reorganize itself to epithelialize the wound and close from the edge to the center of the wound. Larval therapy eliminates infections and biofilms and is a great ally in cases where antibiotic-resistant bacteria are present. Presently, larval therapy is used in more than 30 countries around the world. On this world map, I tried to illustrate, through papers and communications published on the use of larval therapy, the countries that already use this medical device, in blue, and those that do not use it or have not communicated yet, in gray. Our study on the biological behavior of Chrysomia putoria after refrigeration, logistics for use in biotherapy was carried out at the Diptera Studies Laboratory of the Department of Microbiology and Parasitology of the Biomedical Institute of the Federal University of the State of Rio de Janeiro in Brazil. Here is the address for anyone who wants to visit. The first stage of any study related to flies is the assembly and maintenance of the laboratory. Now you go observe the step-by-step of this process that begins with collection in nature. A trap was placed in a high place to prevent the invasion of other insects, with an attractive diet inside, in this case, chicken gizzards. Flies are attracted by their keen sense of smell. They enter through the trap hole but cannot get out. After 24 hours, the traps are taken to the laboratory and flies are identified according to their taxonomy. After identification, the flies are classified, pricked with an entomological pin, and stored in an entomological archive. The egg masses are grown in the breeding laboratory, which we will see below. In the breeding laboratory, the diet with the eggs of the flies was transferred to a 500-degree container, suitably protected by plastic, and placed in sawdust that serves for the larvae to shelter when they abandon the diet. This container with the eggs was transferred to a larger container that was sealed with fabric and rubber bands to prevent the larvae from escaping. The container was identified and incubated in a rearing chamber for the flies to prevent the spread of the bad smell in the laboratory. After the larvae were abandoned from the diet, they were transferred to transparent glass containers, facilitating the observation of biological development. When the larvae reached the pupal stage, these containers were introduced open inside the cage, and as soon as the flies were born, they were offered water and honey diluted by 50% as a carbohydrate source. For the development of the ovaries of the females, 50 grams of chicken gizzard were offered as a protein source. The sterilization of the egg masses was performed according to a protocol previously developed by our laboratory, where 0.200 grams of egg masses were transferred to sterile Petri dishes and mixed with 4 milliliters of sterile distilled water for egg disaggregation. The egg suspension obtained was transferred to a test tube containing 20 milliliters of 2% glutaraldehyde solution, and after 15 minutes of contact, the contents of the tube were filtered through a Wattmann filter paper disc and neutralized for toxicity. A sample of 10% of the eggs was used to perform microbiological sterility tests to evaluate the efficacy of the procedure by direct inoculation in culture medium with soy tryptone broth, TSB, and fluid thioglycolate medium, as described in the 5th edition of the Brazilian Pharmacopoeia. Under aseptic conditions, the eggs were transferred to a surface with gauze moistened with sterile 0.9% saline solution and transferred to petri dishes. At the end of the sterilization process, as previously seen, the petri dishes were sealed with plastic film and transferred in triplicate to a refrigerator with temperature and humidity recorded by a thermohygrograph for periods of 12, 24, 48, and 72 hours. The control was not sterilized or refrigerated and was incubated in regulated climate-controlled chamber at 30 degrees Celsius day and 28 degrees Celsius night, with 60% relative humidity and 12 hours of photophase. The plates were removed from the refrigerator after 12, 24, 48, and 72 hours, and control has been taken away from the climate-controlled chamber. Next, 40 larvae of the first instar were transferred to 50 grams of chicken gizzards and placed in 100 milliliters containers. These were inserted into larger containers of 500 milliliters with sterilized sawdust as substrate for pupariation, and then sealed with fabric and fixed with elastic to prevent the larvae from escaping. All containers were identified and transferred to climate-controlled chamber, regulated at 30 OC day and 28 OC night, with 60% relative humidity and 12 hours of photophase, starting at 6 hours AM. After abandonment of the diet, samples with 5 larvae in the third instar had their body mass determined by means of semi-analytical balances and were transferred to test tubes to observe the development of the larvae until the birth of the adult insects. Each treatment, 3 replicates per treatment, had its respective repetition. The repetition consisted of 8 tubes containing groups of 5 larvae each for observation. The tubes were kept in climate-controlled chamber until the birth of the adult insects. Observations of larval and pupal development until the birth of the adult insect were daily and always at the same time. For the analysis of the raw data, the Microsoft Excel program was used, and the other analyses were used with the R program. Variations in body mass and duration of developmental stages were analyzed using the Kruskal-Wallis test. The viability was analyzed by the proportions test. In all tests, a significance level of 0.05 was considered. The sex ratio was determined as follows: sex ratio equal female divided by the sum of males and females. Now let's talk about our results and let's discuss some issues. The temperature recorded in the refrigerator during the experiment was 9 degrees Celsius maximum and 3 degrees Celsius minimum. Muncoglu and collaborators reported the survival of larvae in first instar of Lucilia sericata for up to 5 days at a temperature between 5 and 8 degrees Celsius without interfering with their viability. The same could be observed in the present study after the process of sterilization and storage of larvae in first instar under temperatures ranging from 9 to 3 degrees Celsius for up to 72 hours. The low temperature storage of sterilized first instar larvae allows them to be shipped to locations far from their original production site. This is because the low temperature slows down the metabolism of the larvae, delaying their development and allowing them to endure hunger for longer, thus reaching their final destination under viable conditions. The maximum humidity during the experimental period was 100%, and the minimum was 19%. Air humidity is important to prevent drying out of eggs and neolarvae. An average humidity of around 60% is ideal, such as that found in the present study. Body mass and mean duration of post-embryonic development stages. The body mass of the third instar larvae that dropped out of the diet differed significantly between treatments 1 and 3. Ferraz and collaborators obtained a mean body mass with values similar to those of this study, as can be observed. Control 0.056 grams, treatment 1 0.056 grams, and treatment 2 0.052 grams, by testing different diets for Chrysomia putoria larvae. This fact shows that, although in our study there was a significant difference in body mass in treatments 1 and 3 about the control, the larvae remained healthy even after the sterilization, starvation, and refrigeration process, reaching body masses equivalent to those obtained in non-adverse conditions, reflecting adequate post-embryonic growth. In this video below, a small sample of some maggot that have undergone sterilization and refrigeration treatment, as you can see, are very active and with size and weights equivalent to those normally bred and kept in the laboratory. The larval development time in treatment 4 was 6.1 days, significantly longer when compared to the other treatments and the control. In the pupal phase, treatment 3 as well as treatment 4 also had a shorter duration than the other treatments and control, with a significant difference between them. On the other hand, the duration of the complete developmental stage, the number of days it takes the insect to complete its biological development, from the larva, pupal stage to the birth of the adult insect, was significantly longer for the fourth treatment, which submitted to refrigeration for 72 hours. Development, larva, pupa, and adults. When the larvae reach their full development, they stop eating and begin to abandon the diet to move on to the next stage of their development, the pupal stage. The rate of feeding abandonment of Chrysomia putoria larvae reached its peak on the 4th day after the beginning of the experiment in all treatments, indicating homogeneity in the development of these insects. Only 8 larvae of the 120 in the control and 17 of the 120 in treatment. One dropped out on the 3rd day after the start of the experiment. Knowing the peak of larval abandonment is critical to monitoring the progress of treatment with larval therapy in the patient. According to Sherman, the larvae remain in patients' wounds for up to 72 hours, which is the length of time they need to consume enough food for their full development. As can be seen in our graph, both in the laboratory and in the wounds of patients, the peak of abandonment has a similar duration. But attention, the development of the larvae can be altered with changes in temperatures and can be shorter or longer. Understanding the duration of the larval stages is crucial for the development of good practices and protocols in the application of larval therapy. This knowledge allows the health professional to monitor the patient, optimizing the change of dressings when necessary, enabling a more effective and gentle treatment for the patient. As for pupariation, the peak occurred on the 5th day after the beginning of the experiment in all treatments, with the exception of treatment 4, which had its peak on the 6th day. The peak of emergence, which is the birth of adult insects, was on the 8th day in all treatments, with the exception of treatment 4, which had its emergency peak on the 9th day. In general, the development showed homogeneity in all treatments evaluated. Regarding viability, there was a significant difference in treatment 4, where 70% of the larvae were viable when compared to the other treatments and control. This probably occurred because the larvae remained without food for up to 72 hours after hatching. The larvae from treatment 4 were also kept at low temperatures, between 3 and 9 degrees Celsius, throughout this period, which may have influenced this lower viability. Even so, 70% of the larvae managed to adapt to the adverse conditions, sterilization process with 2% glutaraldehyde, hunger, and low temperature, to which they were subjected. The other treatments had a viability rate of 95%, as can be seen in the table. Larval viability is the most important phase for larval therapy. This is because the larvae travel a long way in their processing before reaching the application site. Laboratories that commercially produce and distribute larvae for medicinal use generally need to maintain viability for a considerable period of time, since the larvae are highly perishable and undergo a long period of time in the logistical process until reaching their final destination, which it can be in another city or even country, including shipments by air. Stadler 2020 recommends that larvae be distributed and used in the application of larval therapy within 48H of production. Regarding the viability of the pupae, there was a variation between 97.36% and 78.94% in all treatments, with the exception of treatment 4, where only 53.57% of the insects pupetted. This same behavior could be observed in the work of Faraz and collaborators when testing 3 diets for rearing flies, where pupal viability varied from 98% to 71%. Regarding the viability of the insects until the adult stage, approximately 90% of the flies successfully completed their development in the control, treatment 1, and treatment 2 groups. This is important because we can observe that flies that underwent biotherapy logistics were as viable as the control, not undergoing phenotypic changes. In treatment 3, this viability was slightly lower, remaining at 75%. In treatment 4, adult viability was 37.50%, which can be explained by the starvation and refrigeration time, 72 hours, to which the larvae were subjected. The larvae may have suffered desiccation due to being exposed to the cold for a long time. The sex ratio in treatments 1, 2, and 4 was within the expected range, 0.5, based on the formula presented in the methodology indicating stability in the population. Fisher defends that there will only be stability in the population when the sex ratio is 1:1. A deviant sex ratio is not evolutionarily stable because there will be a disproportion in the sex of individuals in future generations. Control and treatment 3 had a lower sex ratio than expected. Although this fact is relevant for the maintenance of dipteran colonies in the laboratory, it is not important for larval therapy. Finally, we can conclude that although the logistical processes for use in biotherapy are quite stressful, the species Chrysomia putoria was very resistant. The flies remained healthy after the entire refrigeration process, which is very important for conservation and transport in larval therapy. 70% of the larvae in the first instar of Chrysomia putoria were able to withstand a period of 72 hours at low temperature, between 3 and 9 degrees Celsius, a fact that allows for more efficient logistics and for a longer time to transport the larvae safely and with quality to distant locations to their final destination, the patient. Thank you all so much for your time and support. A special thank you to Dr. Sherman and Dr. Albert. My contact is Daniele Dallavecchia at gmail.com.
So thank you very, very much. I'm glad we finally had a chance to all watch the video. Whoops, my alarm. And because it's so late, I'm not going to ask any questions myself at this point. I'm going to let the guests or the participants. I'm going to let the participants who've waited so long to ask you questions. Let me put on my gallery view so I can see who would like to discuss this with you.
Okay.
Costa.
Yeah, thank you. Perhaps I missed it. I started also a little bit late, but I wanted to know what is your experience with, what is, you think it is the advantages of this Chrysomia compared to Lucilia? Did you try to use it on wounds? As I told you, perhaps I missed many things.
Estou usando
Sim,
na terapia em feridas, não é?
sim, sim. Porque é que podias estar a usar uma ou outra?
Okay. The experiment was made in Brazil. And in Brazil, Lucilia sericata is rare. Tá certo? Is rare. And for... como é que eu falo por causa disso?
For that reason.
Okay. And... Oh my gosh. Sorry, I'm nervous.
Ok, ok.
Ok, então, no Brasil não há quase Lucilia sericata facilmente, então nós preferimos usar outras espécies,
Okay.
como a Chrysomia putoria.
Okay. From Danielle's saying is that in Brazil there isn't many of Sericata.
Yes, in your state, Rio de Janeiro, it's no facility capturar. Fala aqui, capturar.
It's not easy to capture
Lucilia sericata.
in Rio de Janeiro. So that's why she used the other flies.
Mais fácil essas espécies. Então, no laboratório, a gente criava essas espécies e utilizávamos essas espécies.
So what Danielle is saying, that it's easier to grab those other species so that she can use it in the laboratory. So that's she created, and she used those species more frequently.
I see. Thank you. Thank you.
Okay.
Other questions? Frank.
Yeah, fantastic presentation. Thanks, Danielle. I really appreciate it. And I appreciate that more researchers are looking at the supply chain. So that's very good. I wonder whether Chrysomeria purpura is less cold hardy than Lucilia sericata, and therefore the results you have in terms of poor survival, 70%, at T4, can that be explained because of that? Because they are tropical species and less cold hardy than Lucilia sericata.
Ok, né?
Estou a dizer é que, por facto de teres utilizado aquela mosca, que é dos trópicos, e tem menos acesso ao frio, que conseguiu os 72 graus.
Mas ela ficou em refrigeração.
Okay.
But
The fly.
okay, stay in refrigerations. And the refrigerations about como é que se diz controlado?
Controlled.
Okay.
Controlled refrigeration. Okay.
Understanding? Não. É melhor tu falar. Ou seja, a mosca ficou sempre, né, sob refrigeração, com temperatura controlada,
Okay.
variando entre 5 e no máximo 9 graus.
Okay. What Danielle is saying is that the fly was maintained in a controlled environment, like a cold environment between 5 and 9 degrees.
3 e 9 graus.
Between 3 and 9 degrees, sorry.
Yeah.
Okay.
Okay.
So
Mas aqui, peraí, só completando, mas aqui eu fiz com aquela espécie que eu trabalhava, meu Deus, lembra branca. Calífora vicina, mas não publiquei.
Okay.
Você pode falar para eles?
Okay. Danielle
But here I making this experiment with Calífora vicina, but now publications.
She didn't publish it. Okay.
Sure.
Okay.
that would be great, because I think what several of us are thinking is it would be very nice and very helpful to see a comparison between the different species. We select the species therapeutically and experimentally based on what is most easily available, what is in our area. But it may be that certain species are more effective clinically or may be able to survive transportation or the cold better than other species. And to be able to compare multiple species is so valuable. So I'm glad that you are going to be or have already started testing other species. More questions? Yes, Frank.
Yeah, so you notice that the sex ratio is 50-50 more or less. My question is, what do you think about reducing the male ratio in the population in the laboratory to make it easier for the females to sustain and not being harassed as much by the males? Because you know that males only mate once with a female, or the
Yes.
female mates only once, and then for the rest of the time they're just a nuisance. They're harassing the females. So I think for production purposes, it might be nice to have a lower male sex ratio compared to the 50-50. Do you support that, and how would you achieve that?
I wanted to ask you, Frank, how you think you could separate males from females in a colony? I think that would be extremely difficult. I mean, we know things with tsetse flies that there's done with these sterilization techniques, etc. But well, I think the differences in Lucilia, or I don't know at all, this chrysomia, this was it?
Trisomia putore.
What was your species?
Trisomia putore.
Chrysomia. I don't know whether the differences between males and females are big. I mean, can you separate without problem the male from the female when you look under the microscope?
From this knowledge.
Oh, yeah, obvious.
Ok. Tu pode traduzir?
Sim, sim.
Nesse caso, para esse experimento, né, como a minha intenção era saber especificamente. Vai traduzindo, senão.
Okay, for this case, for this experiment, Daniel's intention was
Sobre a viabilidade larval, ok? Não era muito importante depois o sexo, a razão sexual.
esquecer. Okay, so it was very important for Daniel for the larva viability, the ratio of the sex.
Yes, mas se fosse para a manutenção do laboratório, não é, seria prejudicial.
If it was for the maintenance of the laboratory, it would not be so good.
Mas aí a gente coletava na natureza e inseria no laboratório novas espécies.
For that reason, we collected in the wild and insert them in the laboratory for the new species.
Para tentar equilibrar essa.
To try to set off.
Equalize.
Equalize.
Yes. My focus was viability larval in this experiment for larval therapy.
Yeah. But Costa, to your question, manually selecting males from females is impractical. But there are molecular techniques one could use to make males not survive to adulthood, and then you would have then you would have mainly females emerging.
You are thinking on symbionts or something like that, but they could influence the. Yeah, well,
there's a research project.
I don't think that would be somehow. I mean, for maggot therapy, etc., it might be somehow more difficult, etc., or not really relevant for maggot therapy. And as she said, that she's trying to see the practical aspects of transport, etc., and rather than the negative effects of males on females. By the way, I didn't know that. Can you tell us something more about how you, I don't know, you measure the negative effects of males on females, and how did you do any kind of experiment to see that fewer males, in the presence of fewer males, the females feel better and lay more eggs or whatever? No?
It's something to be done by our students.
Okay.
A student project.
Yeah.
So we have something for collaboration. Okay.
Okay.
Yeah.
Yeah, in this Me Too era, I think it might be good to do something like this.
Yeah. Okay. I have unfortunately to have to leave. I wanted to thank you for your presentation and Daniel, and I wish you a lot of luck and success in your future work.
Thank you, Doctor. Your name is very difficult to pronounce.
Yeah. Just Costa. Yeah.
Costa
Mun-
Muncoglu. Yes or not?
Not
Not.
really,
Muncoglu.
but it's a good
Tell us,
try.
Costa.
Yeah,
Costa.
just Costa, yes. As I told you, Daniel, so just tell me Costa and don't make your life more difficult.
Thank you. Thank you,
so
Costa, for joining us. Thank you all for joining us. It's very late or very early, as the case may be for many of you.
Yeah.
And any future questions, Daniela will certainly be glad to entertain them. She posted her email address. We have that, and we will post that as well. I want to thank you all for joining us and bearing with us and our technical difficulties. So thank you.
Thank you.
Thank
Thank
you.
you.
Thank you. Bye. Thank you.
I will stay on a little bit longer. Daniela, if you want to talk, I know you wanted to talk beforehand. And we spent all that time trying to get the equipment to work, but it may be at the end of your available time. And if you want, we can talk later as well.
Thank you. Okay.
Então está pronto, se queres conversar agora, porque está meia hora antes para conversar com ele. Se queres conversar com ele agora ou queres conversar com ele mais tarde?
No. Agora que me lembro. Não, pode ser mais tarde. No problem, Dr. Shannon. No problem. After, no?
Yeah, okay.
No. After,
Exactly.
for email, I writing better do que falo.
Okay.
Okay.
So we can communicate by writing.
Yeah.
Yes. For me, it's better.
Yeah. Danielle is more fluent by writing.
You're right.
Yeah.
It's speaking no better for me. For in the moments, only in the moments, because I training.
Your video was superb, and that is why I wanted to try again to show the video, because it was excellent in quality. It was excellent in its ability to communicate, and your English was excellent.
My husband helped me in pronunciations.
And that's why we spent the extra time trying to make sure that the video was in the best form that it could be for everyone to see, to hear. So sorry we ended up wasting a lot of time, but I think it was worth it.
No problem. Thank you, Dr. Shannon. For me, it's como é que se diz? Grande prazer.
Pleasure.
It's an honor for me. Stay with you and Dr. Mustafa, Dr. Stade, Dr. Albert, and Dr. Costa. Muglo, and the teachers, Valeria, Alexandre, all people. It's for me, I stay I'm very happy today.
Oh, good. We are happy. It is an honor and a pleasure for all of us. So thank you. And I will let you go now.
Thank you. Thank you.
Thank you all.
Bye. Thank you. Bye. Bye.
Bye-bye.
Bye. 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 Bye.